Well, good evening, everybody. Thanks for attending this evening. Just give it a few more minutes. We'll see if we can get as many people in before we start, and then we'll get cracking. It's not going to be a very long talk today. I wanted to give you the chance to ask as many questions as you might have. Just as a bit of housekeeping, I'm not going to be using the chat function. I'm going to be using the Q&A. So there'll be a little Q&A box on your action bar. You just click that and you can write your questions in there, and I'll go through them at the end, and we'll go through as many as there is. Right, just a reminder, everyone, please use the Q&A function to ask stuff. Don't put it in the chat, just so that I can keep everything in one place. Right, another minute and then we'll get started. I think we're giving everybody enough time. Okay, right, let's get started. So I wanted to do this webinar tonight just to give everybody a little bit of an update, I suppose, about the Varroa mite crisis in New South Wales, but specifically in relation to the native bees. So this isn't going to be a talk about the honeybees or how the Varroa mite is going to be affecting them, beyond the explanation of how the Varroa mite works. Anyway, so I'm going to be focusing on native bees. So if there is anybody listening who has a honeybee hive and they're more concerned about that, what I would suggest is get in touch with the DPI directly about that, because you can ask anyone's opinion, but ultimately the DPI will be the ones that dictate what happens. But this is in relation to the native bees and the sort of the knock-on effects of this crisis here. So everyone's probably heard of the Varroa mite. But up until very recently, up until this recent incursion, Australia was the only country in the world that didn't have it. So up until now, we've been doing quite a good job. But it has obviously arrived now. So I'm going to talk about first a little bit just about how the Varroa mite works. So its life cycle, what it does, why it's dangerous, why we're worried about it in terms of the honeybee industry, and then we'll cover a little bit about the incursion that's happened here in New South Wales, and then at the end we'll touch on what it might mean for our native bees specifically. And when I'm talking about native bees in this context, I'm referring almost primarily to our stingless bees, the Tetraconula carbonaria that we have in Ku-ring-gai. If you've got any questions in relation to other native species, we can look at those. But the reason I want to focus on the Tetraconula is A, because of course it's a key part of our council programs, but also it's probably the most at-risk native species, native bee species that we have in relation to this. So, so what is the Varroa mite? How does it work? So the Varroa mite is it's an obligate external parasite of the honeybee, basically. So obligate, it simply means that they can only infect honeybees, right? Their life cycle is reliant on that of the honeybee. And you might be thinking to yourself, after I've said that, then why is there a danger to native bees? And we'll get to that. But interestingly, you think, well, hang on a minute, these mites are obligate pests of the honeybee, so why can't the honeybee seem to cope with them? You would normally think that in a system where that's their natural host, that there'd be some sort of resistance. Well, the main reason is because the Varroa mite originated in Asia with the Asian honeybee, so Apis cerana. And they actually do have methods for coping with Varroa mite, various behaviours that they employ to prevent the mite from doing any significant damage. Now, of course, most of our commercialised bees are actually Apis mellifera, or some subspecies of, so the European honeybee, and these did not co-evolve alongside the Varroa mite and so are particularly vulnerable to it. So what the Varroa mite does is it feeds on the bee's blood, basically, well, or in what the equivalent of bee blood, which is hemolymph, and that's what they feed on. So they are, essentially a blood-sucking parasite. Now, this problem of Varroa mite, it's perhaps unsurprisingly, is human-induced. It's our fault. It's occurred because of the movement of European honeybees into Asia, and then the subsequent shifting of host of the Varroa mite to the European honeybee. And there's two sort of what we call haplotypes of Varroa mites, two different sort of regions where they came from. We have the Japanese and we have the Korean. But either way, they jumped host to the European honeybee when we were moving them around for agricultural purposes. And now they are pretty much ubiquitous across the globe, apart from here in Australia, at least until very recently. And they are the world's most devastating honeybee pests. So they cause the most damage, and both in terms of the bees themselves, but of course the knock-on economic effects of those. And I think that's worth stressing here as well. So we have to remember that we're looking at these bees, and the honeybee in this case, as what it is, which is a domesticated animal. Right, so we're not thinking about it as a wild animal, we're thinking about it as a domesticated animal. And that's very much how the approach to dealing with it has been looked at. It's being looked at as an economic issue, really. It's not really being looked at quite so much as an environmental one. So this map here just gives you a good idea of where the Varroa mite is. It's pretty much everywhere. So where it's red is where the Varroa mite is. The grey areas are either countries where we don't have any records, so you might spot North Korea there. I can't imagine they were particularly open about it. And some of the places in Africa are in grey, mainly because the Africanized, or the African honeybee, not the Africanized, my apologies, the African honeybee does show some resistance to the Varroa mite. So they're in grey. But all the red areas are the areas that are predominated by the European or Western honeybee. And so you can see they are everywhere, from the Americas through Europe, Russia, China, Asia, and New Zealand as well. So Australia there being the only country that hadn't had it. So as a little bit of, this is a diagram of the life cycle. And if you want to sort of read those bits, you can, but I'm going to summarize it sort of quickly. So the Varroa mite's life cycle all takes place inside a honeybee colony. So it's completely contained within the honeybee colony, and it is reliant on the way honeybees reproduce, which is why the Varroa mite doesn't directly infect the bee species. So essentially an adult female mite comes into the colony on an adult bee, perhaps from a drone, which is quite a common way, or through a worker, and she finds her way into an open brood cell, And when she's in there, she lays her eggs. So she first lays the male, and then she lays some females. The male hatches first, and then the females hatch afterwards. They mate inside the brood cell. The male dies, and then the females begin to feed on the pupa, pre-pupa, and the larva inside. Now, what then happens is, if it's a drone cell, for example, that takes a lot longer to develop than a normal worker, they can actually reproduce multiple cycles within that one brood cell. And then essentially the females, when the adult bee hatches, or perhaps if they're fed on it to such a degree that it dies, then they leave the cell and they're attached to other adult bees, and they can spread through the colony that way. And the females are very mobile as well, so they're able to quite literally walk across and through the brood in order to spread themselves. And then mite transfer occurs through the touching of individuals. So it can spread to another colony, for example, by bees from different colonies interacting with each other. Perhaps that's through the raiding of a colony, perhaps it's through drones going from different colonies. But you can see how it can sort of latch a ride and find its way, to the next colony. Now, their growth in the colony tends to be exponential. So actually, with Varroa mite, you can have Varroa mite in an infected honeybee hive for a number of years, actually, before you may detect it, because they often start off in very small numbers, but then they start to really exponentially grow as they continually reproduce within there. And that's also linked to environmental conditions as well. So in colder regions, the mite cannot produce quite so prolifically. So therefore, it's harder for them to produce these huge numbers. Whereas in the hotter areas, the warmer areas, where the honeybee produces brood all year round, unlike in cold areas where in the winter they tend to not have any, then that means the Varroa mite can essentially reproduce all year round as well, and so can increase very quickly. So actually, here in New South Wales, where we still get the sort of the coolness in the winter, there's perhaps a benefit for us in order to help control it. If this infestation had occurred in northern Queensland, for example, then actually it would have been a lot more difficult to try and contain. So what is the impact of it? Why is it so bad? So we've spoke about how they infect the colony. They get in there, they suck the blood of the hemolymph of the developing bees, and then they spread via the adults, and they go to colonies. But why is that a bad thing? I mean, you could argue that there's lots of animals that might have an external parasite every now and then. The problem with these is, because the honeybees tend to have very little behavioural adaptation, they proliferate very quickly. But also, they're drinking the hemolymph, the blood of the young bees as they're developing. So they're taking that energy and all the nutrients away from them, which means that they often don't grow properly. So they can be stunted, much smaller than they would normally be. They can live a lot less. They can struggle to fly. Research has shown that it affects their memory as well, so they struggle to find the way back. If it's a particularly heavy pest load, it can kill them outright, even before they become an adult. But actually, one of the real sort of dangerous part of Varroa isn't actually the mite itself in terms of the physical damage that it does. It's often the knock-on effect that has. So the mite itself also carries a number of honeybee diseases, and it can act as a viral vector. So it can crawl throughout the colony and spread that virus throughout the colony to the rest of the bees. But also, it makes them more vulnerable to any other infection that they may have, or that may be sort of dormant inside their body. So often a lot of honeybees carry what we might call silent viruses, where they're sort of at a very, very low level, and they're not particularly virulent, and they exist at, and sort of tick along in the colony, and they don't really do any damage. But when you've got Varroa in there, it is suppressing their immunocompetence, so it's making them less able to deal with infection. That can then cause the virulence of the other viruses, either spread by the mite itself or that happen to be dormant in the population, suddenly explode. And so the picture on the left of the screen there is a honeybee with deformed wing virus, which is a commonly linked virus to Varroa. And we see that hives or areas that are particularly heavy in Varroa infestation also have higher levels of other infections too. So it's this huge knock-on effect that the Varroa mite has that makes it such a dangerous honeybee pest. It's the fact that not only does it physically damage the bees and it can spread viruses itself, it makes them far more vulnerable to everything else. And so ultimately this can lead to the death of the colony. You lose workers, your workers aren't being replaced, and eventually the colony collapses and dies. So that gives you an idea of what the Varroa mite does. So from that you can understand perhaps why the honeybee industry and honeybee keepers generally, whether they be commercial or hobbyist, are so worried about this. The other part as well is that Australian bees have never been exposed to it before, so we're particularly vulnerable to an infection because, unlike the rest of the world where they've had it for 20 years now, we haven't, which means that our population is likely to be quite vulnerable to it. Right, so let's just get on to the New South Wales crisis and what's happened. So it was originally detected in Newcastle at the port there back in June in 2022, and it was detected in one of the surveillance hives. So the DPI has surveillance hives at all the major ports, and these are used specifically for these reasons, to detect when things might come into the country. Now, officially, we don't really know how this happened yet. It's possible that it came in through the illegal importation of an infected queen or queens, where someone essentially smuggled them in to Australia. It could have come from a swarm or something similar that was on one of the ships. I think the former is probably more likely, given the way that it spread, but fundamentally we don't know. And as far as I'm aware, the DPI hasn't released any information on how the infestation first started. Whether that's because they don't know or because they don't want to divulge that information, I don't know. But suffice to say, it happened. We arguably had let our biosecurity slip a little bit. So up until 2015, our sniffer dogs were trained to sniff for bees for the reason of people trying to smuggle queens. But for a period after 2015, we didn't train the dogs to do that. They weren't sniffing out these feral bees. That includes the time in which the incursion happened. So I'll leave that up to you guys to decide. But I think maybe we got a little bit lax, thinking that it was never going to arrive. So it landed in Newcastle, and the little map there in the left just shows you what it was like after the first couple of weeks. So you can see the red spots where infected hives were found in Newcastle itself, and then one a little bit further north. But now it's spread much further than that. So it's spread as far south as the Central Coast. So actually, where I'm sitting speaking to you now, I'm in a red zone. So I'm in Woy Woy on the peninsula, and that is a red zone. The red zone now extends across the entire peninsula down to Mooney Mooney near the Hawkesbury. And so it's creeping south, and it's very, very close to Ku-ring-gai now. In fact, the surveillance zone, the purple zone, which we'll get onto into a second, which is put around these red zones as a sort of buffer, encompasses the majority of Ku-ring-gai now. So we're very, very close to having a potential infection. There's a couple of isolated infections as well at the north coast, Narrabri's not north coast, but it's up north, and Coffs Harbour on the coast. Now, they occurred through the movement of colonies from the infected area in Newcastle to those locations. Now, we don't know officially whether that was done before the restrictions were put in place or not. I think they probably were, but we don't know for sure. But look, essentially, the DPI have adopted an eradication approach, this idea that they're going to try and eradicate the mite, get rid of it completely. But that was since June 2022. It's now mid-April 2023, and frankly, the mite's still spreading. Maybe not as fast as it would have done if we'd done nothing, but it is still spreading. So this is the map now. Well, it doesn't include the Coffs Harbour bit, but it includes the bit around where we are. So you can see now it encompasses all of Newcastle, a significant portion of the Hunter, and you can see that red blob down the bottom there covers Central Coast, so it covers Gosford, Somersby, the peninsula, and you can see it's very, very close to Varroa just in the bottom there. So, you know, all of you now sitting in Ku-ring-gai, Varroa is not far away. So, you know, that gives us a really good idea of how close the mite is. And it's worth stressing here that you know, one of the dangerous parts we're in now is that with this central coast infection is that, of course, there's a huge amount of national park between the central coast and northern Sydney and both all around that area, which means, you know, there's not a lot of monitoring that goes in there. So things like feral colonies, which is what we're going to talk about in a second, become the real dangerous aspect of the spread of the mite, the things we can't control for. Right, so I just want to explain the exclusion zones, because these are what the DPI had put in place in relation to the Varroa mite outbreak. So the DPI's management plan essentially utilises 3 zones. So you have a red zone, which is where an infected hive has been found. And then you have a 10-kilometre radius around where that infected hive is, and everything in that 10-kilometre radius is destroyed. So all the managed hives are destroyed, irrespective of whether they have Varroa or not, they're all destroyed. And then they deploy poison bait stations in those locations in order to try and get rid of feral colonies, because we don't know where they are, right? And it's these poison bait stations that are, pardon me, the core threat to our native species, and we'll discuss that in a second. So, and then outside of that 10-kilometre zone, there's the purple zone, which we call the surveillance zone, and that's a 25-kilometre radius outside of that red zone. Right. And basically within that, that's where everyone's on high alert. So the DPI will be inspecting managed colonies. If you're a beekeeper, you have to have reported your beehives to the DPI. That is a legal requirement. And also within this purple zone, if we happen to find any feral colonies out and about, we must also report those. Right? So that includes us in Ku-ring-gai, because we are in a purple zone. So if you've got a honeybee hive and you've not reported to DPI yet, you need to. And if you know the location of a feral colony, you need to report that to the DPI as well. And that's actually one of the core things that we can do, and that'll be the point that I finish on in detail. And then the blue zone is the zone everywhere else. It's the rest of New South Wales. So this is just essentially, there's restrictions in what you can do to move colonies. And if you do want to move a colony, you have to apply for a permit to do it so that we know where they're going and that they can be tracked properly. So essentially, that sums up the different restrictions depending on the different zones that you're in. Okay, so the DPI have adopted an eradication approach. Now, there's mixed opinion on to whether this is possible or not, I think, or feasible. So a number of academics have suggested that, given the number of infected properties and the spread, it's probably quite unlikely that we're going to manage to eradicate the mite. It's also worth mentioning that no other country in the world managed it, although I would argue that we are the best prepared of any other country, simply because we're the last ones to have done it, and we have, you know, we've put things in place beforehand. But either way, that's the approach the DPI is still going for. I mean, you know, this was started in June, and it's now, you know, coming on to a year, and we're still getting new detections, not as many, but we're still getting them. With any type of eradication approach, you need to hit hard and you need to hit fast. So if we're still seeing them coming up now, you know, perhaps there's an argument here that the eradication method's not working, but it's certainly the way that they're going with it at the moment. And as we discussed, that means that all hives within a 10-kilometre radius of infected premises are destroyed. So that's all the managed colonies, and then they move to try and destroy the feral colonies. And it's this destruction of the feral colonies which is the big sort of risk for our native species. So essentially, the challenge that the DPI has with feral colonies is, well, look, we know there's loads of them out there. I mean, I don't think it's too bold to say that, generally speaking, we've been pretty rubbish at controlling feral honeybees. They kind of sit in a, we don't see them the same as things like rabbits and foxes that need to be, you know, culled and controlled, even though they are the same thing. They are an escaped feral domesticated animal, and you know, more and more evidence is showing that they do damage native plant pollinator systems. And so they are something that we should manage, but we just haven't really. And so now, we have so many feral colonies out there, most of which we don't know where they are, not managed by a beekeeper. So they're not, in a managed hive with a beekeeper, they are constantly keeping their eye on the colony. They're checking to see if there's something wrong. Are the bees not well? They can check for Varroa mite. They can do all these things, right, to keep the honeybees clean. In a feral hive, none of that's happening. So these feral hives can become huge parasite sinks where you can eradicate all the managed colonies in an area, but if you don't also get rid of the ferals, then the pests just sit there. And then when you bring the colonies back in, new ones, they just get infected again. So it's a challenge, because what do you do? Because you don't know where they are. So what the DPI have done is they use poison bait stations. So they rely on the behavior of honeybees, which is that they will raid colonies, dead colonies. And they use a pesticide called fipronil, which is a broad spectrum insecticide, which is particularly toxic to hymenopterans, so bees, ants, wasps. And what they do is they set up a bait station, and they wait until they get a huge number of honeybees coming to take the sugar syrup, which they use as a bait, and then only then do they add the poison when they've got a high level of recruitment. And then those stations are manned by a DPI staff member the entire time that the poison's in there, and then they'll remove the poison when there's nobody there. So there's only a brief window that it's there. But what happens is these feral bees, theoretically, will come to these stations. They'll gather up this poisoned sugar syrup, and it's a slow-acting poison relatively, so it's not going to kill the bees straight away. They take that back to their feral colony. It builds up inside that colony, and then it kills that colony. And that's how you eradicate the ferals, right, without knowing where they are. Now, the problem with those is that, well, does it affect our native bees, right? And I think we've mentioned before that the Varroa mite itself does not and cannot infect native bees. The life cycles just don't match, and the Varroa mite is tied in very, very tightly to the life cycle of the honeybee. But there is evidence to suggest that honeybees can pass on viruses and diseases that they've caught to other native species. It's been shown in bumblebees, for example, in European countries, in North America. So the Varroa mite is something that we want to eradicate for the safety of our native bees too, in many respects, because what we don't want is there to be a huge, disease load in our honeybees now, and then they start passing that on to our natives, which won't get the same level of treatment or management as honeybees will. But the direct sort of threat currently with the eradication plan is the threat of secondary poisoning. So this idea that either the bait station attracts stingless bees mainly, who are just as vulnerable to fipronil, in fact more so, because they're a fraction of the size of a honeybee. So the dose they're getting, per their body weight is much bigger. But also perhaps the more insidious threat, and the one that's probably more likely and the one that's more difficult to control, is that stingless bees will also raid dead honeybee hives for resources. Now, if you are using a bait station to poison feral honeybees, and you've got somebody there so that if tetragonula start coming into the bait station, they'll close it. So the DPI won't leave it open. They will close it because there's a risk to native wildlife. So it's not that they're out there to scorch the earth or whatever. But the problem is that even if you're not getting the tetragonula go into these traps, what you will have to some degree is you'll probably have all these feral hives sitting out in the bush, no one knows where they are, chocked full of contaminated honey. Bearing in mind that this fipronil can stay active for nearly 3 years in the bush, right? So it's not like it's just going to evaporate after a few days. It can stay in the bush and be toxic for a very long time. So the threat here is that our bees will end up going to these feral colonies. They'll take these resources out, they'll bring them back to their own colonies, and it'll poison them and kill them. So that's the real big worry with our native bees. And the problem with it is that we're very much we can't control for it because we don't know where the ferals are. So you don't know if your colony is going to be near a feral hive or something like that. So that's the concern. That's the idea of what the threat is to our native bees immediately. Yes, things like the knock-on effects of disease transfer from honeybee populations to native bees later on down the track, if we don't eradicate the pest, is certainly there, and that's something that we will be considering in the long term. But in the immediate future, what our worry is of this secondary poisoning. Right. So now this is the last little bit of the talk, so I don't want to go on forever. But this is now stressing what we can do about it. I wish there was more we could do about it. I'm afraid there's scant little that we can do. But as I mentioned, the feral colonies are one of the key challenges in managing the Varroa mite and in also the threat to the native bees, because we simply don't know where they are and we don't have any control over them. So because these feral colonies are not only sinks for the mite itself, so they can then secondarily infect other colonies from them, they're quite ubiquitous, to be honest. Some of you listening now will probably have already seen a feral colony. I've seen feral colonies through the Brisbane Water National Park, quite common. So you sort of got to think, if you're spotting a few on some walking tracks, I mean, how many are out in the areas of the bush that you can't walk to? Probably lots. So these are the danger. Now, even if you were to destroy all the managed colonies in an area, if you've still got ferals there, you're still going to get a problem. Now, the major thing that we need to do is we need to report feral colonies. So what the DPI are woefully underfunded and they're woefully understaffed, right? They have a huge challenge on their hands to try and deal with this. And what they need is they really need help from everybody, okay? And we know our local areas better than the DPI will, okay? So, you know, we go bushwalking, we know those areas in our backyard, right? So if we know of a location of a feral colony, even if it's a historic one that you sort of remember from way back when, really, really, really important that you report that to the DPI. Ideally with a GPS location, but at least a street address, if not. And you can use that phone number there to report it. Or you can go online, there's an online form on their website that you can fill in as well. Because if the DPI know the location of a feral colony, they can go and destroy it. They don't have to rely on a poison bait to do that. Now, I'm not saying that if we report all these ferals that the DPI aren't going to bait in an area if there's an infection. They will still bait. Even if by some miracle we manage to find every feral colony in Ku-ring-gai and we remove them all, they will still use these poison bait stations. But the important thing is if there's less feral colonies out there, we're going to have less of these dead colonies that are sitting in trees that we don't know about, that are a big puddle of infected honey that might be secondarily poisoning our native bees. Because there are, as I mentioned, you know, the DPI do have measures in place to prevent our native bees accessing the bait stations, because they're constantly manned, there's somebody there. But they can't control whether they raid a feral. So if we're reporting as many ferals as we can and they can be destroyed directly, then it means that even when the baiting stations come in, there's less likelihood that a feral colony in the area will need to be destroyed that way, which means there's less likely to be these big sinks of contaminated honey in these areas. So that's what I really want to stress to you all today, that that's the thing that you can do as a member of the public to really contribute and help. And I've already had a number of residents who have said, oh, I know where one is, and they've taken a picture of it and they've got a GPS location and they've sent it into the DPI, or if they've not been able to do that, they've sent it to me and I've done it. And it might seem like a drop in the ocean, but I implore you to do it, because it really could make a difference in a local area, especially because our little native bees, they don't forage very far. They only forage 500 meters roughly from where they live. So that means that even if you're in an infected area, if there's no feral colonies in your 500-meter, sort of kilometer radius around your colony, then actually you've got a really good chance of your bees being completely fine, right? So it really is something that we can do. So I'll go through your questions now, and please ask as many as you can. If there's any that I can't answer, or I'll either find out, or I'll point you on to a resource that will answer that question for you. So let's just open up the Q&A, and I'll start from the top, and I'm going to move my way down. Jacqueline asks, have you seen or know of a Varroa attack in the Great Sydney region? No. So we rely on the DPI's interactive map, which you can also find on that link below, and that tells us where the infected premises are. There has been nothing recorded in Sydney yet. I do stress the yet. I suspect there will be, but I remain hopeful that we can avoid it. Look, there's a couple of quick questions on here about, the stingless bees in relation to the program. If you've got any questions on that, please just email me, because that's not the topic of tonight. So I want to focus on the Varroa mite stuff. So Helena asks, how do other species of bees manage the mite? Could any of these methods be applied to apis mellifera or native bees? Yeah, so for instance, if they were bred. So the answer is yes. So there are ways, and many countries that have had Varroa mite for a long time have been looking into this sort of idea of well, how can we, we've got to cope with it, right? So how do we do it? So what we find is that there's actually quite a lot of variation between colonies, even of apis mellifera. So if we exclude the Asian honeybee, which has evolved with the pest and is able to cope with it, even within the Western honeybee, we find that there are certain colonies that employ more hygienic behaviours. So they clean the mites off, for example, or they clean out infected cells. And what a lot of beekeepers have been looking to do is to breed from that stock. So often what happens is, you may lose a number of colonies in an area to Varroa mite, but a few of these colonies, although they were infected, they've still survived and they've come on. So that implies that they've got some type of adaptation or perhaps some sort of resilience to the mite. And so people are trying to breed from those resilient stocks. We still don't really know necessarily the ins and outs of what it is that is making those bees more resilient to it, but we are finding that actually there is quite a lot of heritability to it. So you can essentially breed that. So yes, there are ways that we can do that. We haven't been doing that here in Australia, despite the fact that it's been done elsewhere. I think there's mixed reasons for that, but it may be something that that happens going forward, because even if we do get rid of the mite, people may decide that it might be a good idea to try and breed in some more of these hygienic behaviours. There's also methods you can apply as an apiarist as well, but I won't go into those right now. Okay, so Peter asks, if our native bees are ones that just stay in and around our hive, how does the pest get into our hive? Are there any other travelling species that visit our hive? So, right, so as I mentioned, the Varroa mite doesn't infect the native bee hives themselves. That's not how it works. So what's the danger is actually from the way we as people are trying to deal with the Varroa mite, because we're introducing a poison into the environment, and it's actually that poison that can be vulnerable. However, there are other pest species that affect stingless bees. The three main ones would be small hive beetle, which again is primarily a honeybee pest that's introduced, and then there's two native fly species, the circuit fly and the forehead fly. So our native bees certainly have their fair share of pests, but none of them impact a population to the same extremity as Varroa mite does to the honeybees. And so essentially what happens, the way the mites are traveling is they're hitching a ride on the honeybee workers or on the honeybee drones, and then they're spreading to other honeybee colonies that way. Yeah, so this person anonymous, but they've asked a really good question. So it says, as ants are also exposed to secondary poisoning, what would happen to poison feral honeybee colonies in that relation? You're exactly right. So it's not just native bees that are at risk here. Any native hymenopteron that may be involved in the raiding of the contents of these feral colonies would be at risk. So both ants and wasps would be at risk there. So a plethora of our native wasps that would also raid a dead honeybee hive for the honey will almost definitely be killed. And ants would be the same, and it would work in exactly the same way. So the workers would bring that poison back to the colony. They share food between each other, and so they essentially spread the poison amongst themselves, and that's how it kills them. So I've focused on the native stingless bee here today, but it's by no means the only species that's at risk of knock-on effect here, and there are lots of people that are calling for the halt of this eradication policy because of the risk of knock-on effects to native wildlife. Yeah, so someone's always said, that the stingless bees tend to rob for propolis rather than honey, and so the risk is minimalised. Yes, they do rob for propolis. No, the risk is not minimalised because the fipronil will also be in the propolis as well. And it will be in the propolis to lesser degrees, but because the stingless bees are so much smaller, they won't require a huge amount of dosage either. So the risk is still there. So actually, yes, they do tend to go for propolis more than they do for the honey, but the risk is very much still there that fipronil will have infected the whole colony. So Tom Sheepy said, when you talk of feral colonies, you mean only European bees? Yes. So they're the only, well, technically Asian honeybees would also be counted as feral honeybees, but we have quite a strong control measuring for those, and they have not really established themselves. I think there was an incursion up in northern Queensland a few years ago, but they're under quite a strong control measure, mainly because they can bring Varroa mite with them, which was one of the major reasons why we have a control measure for those. But yes, in terms of feral colonies, because they're the only non-native bee that builds a colony, technically they're the only feral bees that you can have. So Lindy asks, how's it managed in Europe or the USA? So it's managed in Europe and the USA through a combination of miticides, so specific sort of arachnicides that are aimed at killing the mites. So they treat the colonies with these miticides and control for the mites that way. There's also ways of managing your colony to reduce mite numbers. So, for example, because they reproduce in brood, in areas where, cooler areas where the honeybees sometimes don't stock any brood at all, what a honeybee beekeeper can do is manipulate that by putting in essentially a tray of brood, which draws all the mites into it, and then they can remove that and destroy it. So that helps keep the numbers down. It's often a combination of these methods. So what we would call integrated pest management, this idea of mixing husbandry techniques and chemical controls to try and manage these ways. But of course, none of it's perfect because that all takes a lot of extra time. It costs a lot of extra money. So it means there's all those knock-on effects to the honeybee industry in terms of the increased costs and the decreased output. You've also got to be careful with what chemicals you use, because of course, if you're selling your hive output, which is what you want to be doing, your honey and your wax and things, you need to make sure that there's not being any chemical contamination in those as well. So it's certainly, prevention is certainly what you want. You really don't want to have to be trying to manage this. And as I mentioned earlier, there has been some success in breeding these more hygienic traits into genetic lines so that the bees are more able to look after themselves as well. Helen LeGras says, is there any reason to preserve feral hives? Really good question. I think it depends on what side of the fence you stand on. So if we're talking about environmentally, no, they don't belong there. They do a lot of damage, and actually removing feral honeybee hives would be good for native bees because they outcompete a lot of our native species. So to remove feral hives from the bush would mean that our native species would have a much better chance of re-establishing themselves and getting their numbers back up again. There is an argument that in certain areas, especially areas that rely heavily on pollination for agriculture, that these feral colonies contribute to that. And because many of these areas have been highly degraded in their natural environment, they probably don't support a very strong population of native bees anymore because they've been so badly degraded. Look, I'm always, from my perspective as a native bee scientist and someone who is interested in conserving, I really think feral bees should be eradicated, or if we didn't have the Varroa mite problem, then feral colonies can be moved into managed colonies so they can be managed by a beekeeper, and then that way we can keep them in the areas we want them to be in and not in our natural areas. Peter says, that's a long web address. Can you email all the contact information? Yes, I can. So we can send out an email with all the bits and pieces on at the end. Bearing in mind this is being recorded, so that's all going to be sent out as well. So you'll all be able to get all the stuff you need to do. Also says, if the DPI destroy a feral colony, is that no longer a problem? Essentially, yes, because what they can do is when they destroy a feral colony, they can remove the infected material because they know where it is. It's sometimes more difficult because it's not in a nice managed box. And I suspect that there may be some scenarios where that's not possible, depending on where the hive is located. But most of the time, they should be able to remove that material. But also, they don't have to use fipronil to do that. They can destroy it in more traditional methods that don't leave behind residue for native bees to use. So that's why that's a really good thing if we can locate where those ferals are. Is there a simple app that helps pinpoint exact GPS if we see ferals? I don't know about an app that does that, but I find Google Maps works quite well. I use that when I'm out and about if I need to pin drop something. So you can always try that. You know, saving that if you can't and it's on a track, you know, then the best thing to do is take a photograph of where the colony is and then a photograph a bit further back so you can get an idea of the surrounds, and then just you can explain, you can write in the form, or you can speak to DPI on the phone and just say it's down Darry track and it's, you know, approximately half an hour or it's approximately 15 minutes down here. Anything that you can offer is better than nothing, because up until that point we had no idea it was there. Right, Goldie asks, are there any obvious signs of secondary poisoning apart from lower bee activity? No, not really. The problem is that it's because it's quite insidious, you probably won't know until it's too late. That's why it's worrying. So even the DPI themselves have suggested that for people who do own stingless beehives, that they can, if they can, to move them out of the red zones if the red zone is there. Of course, for our program, that's impossible. We simply have too many colonies. But for those people who have one or two, then that's a possibility. But no, pardon me. Unfortunately, you wouldn't really know until the bees started dying, and by that point, it would most likely be too late, because it can destroy a colony within 48 hours. Margie, would DPI use what 3 words, since that's easy for most? I don't know, you know, that would actually be very good, wouldn't it? What 3 words? I don't know is the answer to that question. Again, the best thing to do may be to ring the DPI if you want to know if that's something they use. So Helena Grice asks, how are feral hives destroyed? Well, there's a number of ways that it could be done, and I don't know exactly how the DPI would do it. It's often done by people who specialise in feral colony removal. So actually, we work with a number of people who would remove feral colonies alive and place them into boxes. However, they won't be able to do that with Varroa mite. There's a number of different ways. You can gas them, for example. That may be one way they do it. I think it would probably be quite location specific, and that's a question that you'd have to ask the DPI, I think, because they'll have their own policy on how they're going to do it. Caroline asks, does the DPI have any plans to reimburse any native hive owners who have hives that die because of the fipronil baiting? Honeybee hives need to be used. Yeah, as far as I know, no, Carol, they don't. So I think they're trying to cover themselves by saying that because the stingless bees are not covered by the emergency order, you're allowed to move them. So they advise people to move them. And they may claim that that's why. I'd also imagine you'd have to prove it, which is quite hard for an everyday person, because how do you prove it? You'd have to have it tested at a laboratory. So yeah, I think you would struggle to get any reimbursement for that. Look, I mean, if it does move into northern Sydney, I think the DPI is seriously going to have to consider that it's now too late. They're going to have to, opt for a different different method. But yeah, I would say there's not a great odds that that'll happen, unfortunately. So another question is, how can DPI effectively, I think that must mean reconstruct the movement of honeybee colonies, given the demand for pollinators by growers of almond and other bee? Well, it's a brilliant question, isn't it? Because yes, what do? You've wiped out all of your honeybee colonies, right? You can't bring them back for 3 years because you've got the fipronil bait stations out there. And the research has shown that if you bring honeybees, clean honeybees, into an area that's been baited in the first 12 months, a lot of them die anyway because they get secondary poisoning from foraging from the feral colonies. So what do? Well, I feel a little bit like I'm Mr. Hindsight here, but what we should have done is not rely on one pollinator. But that's the big mistake that we've made in Western agriculture generally. We've put all our eggs in one basket, and now we're going to pay for it. Because really, like any system, even if we think about business or trade, diversification is what you have. If you have diverse assets, it means if you have something goes wrong with one, you've got other ones to fall back on, right? But we've ignored that. And so we've relied purely on the honeybee, and now we're scrabbling for replacements, right? And it's why the stingless bees have become so popular recently, because they are a great alternative to honeybees. But it's a good question, and I think, to be honest, there isn't really an answer for it. I think, especially for things like almonds that are very, very dependent on honeybees, if they were to lose colonies in those areas, there would be a severe impact on that industry. And I don't think there's a very good answer for that question. I think it's more worrying. Think about that. So, right, Andrew Will says, will the coming winter slow the Varroa mites or kill them? Yes, it will slow them. So they can go into a sort of, it's not a hibernation stage, but it's sort of a reduced stage of activity. They could survive for, I think it's about 3 months on an adult bee. So that will certainly slow their spread. And so now that we're coming into winter, that's also helpful. It will also slow their spread because the honeybees move less in the winter months. And they also, I would imagine in many areas of New South Wales, and where there would not be infections now, they also won't produce anywhere near as much brood during those colder months. So I mentioned earlier that the odds of eradication are quite low. Having said that, now we're coming into the colder months, it may give us the edge we need to get on top of it, because there's not going to be as much movement there. Goldie asks, if a bait station is being managed near my hive, can my hive be moved to a different area? Yes, it can. So if you've got a stingless bee colony, you can move it wherever you want in the state. So I would encourage people that if you do own your own, and you've got a friend in, I don't know, Byron or whatever, then consider sending it there on holiday. Bearing in mind that if we do get a red zone in Ku-ring-gai, then they'll be at risk of poisoning for up to 3 years. So the hive will be away for quite a long time. So, and then there's risks of having someone looking after it who doesn't know what they're doing. And look, there's risks there. But yes, you are allowed to move your hive away if it's a native colony. Goldie, what can we do to encourage Katie to tackle the issue of feral hives as I do? Me. That's how we do that. So I'm already quite active in that space now. So I'm working with our operations team and with our current staff that work on feral pests to begin to include feral honeybees in our pest reporting. And in the past, before the Varroa mite came, even before then, because I was interested in getting rid of them from a conservation perspective, we were having a number of whenever we found colonies, they were being reported to me, and then we were having a beekeeper come and remove those colonies and then put them into a managed hive and take them elsewhere. So we have been active on that front, perhaps more so than many other councils. This Varroa mite issue has only pushed that to the forefront. So I've already prepared a number of materials for council staff in order for them to understand what they need to do with the Varroa mite problem. And I hope in the future that we will be treating it more like foxes and rabbits. With the Varroa mite, we'll have to kill them, but when that leaves, there's no reason we need to kill the feral hives. It's not what we're after. We just don't want them in the bush. We just want them to be in managed colonies in areas where we know they can be looked after. Yeah, so I mean, just a simple statement. It says, you know, feral colonies compete with birds and mammals for nesting space. Exactly. I mean, there's probably a few of you on here who might have seen the odd possum box or something like that taken over by a feral honeybee colony. They will turf out birds from their nest too. So there's a number of reasons why we should manage them. Lindy asked, what is council doing to protect native beehives in KRG? It's a good question. So we have a particularly large challenge because we have so many colonies that it will be impossible for us to move them because we simply wouldn't have anywhere big enough to do it. So what we're doing is, we're being proactive. So we're looking to find feral hives and feral colonies early, remove them so that the risk to Ku-ring-gai hives secondary poisoning is reduced down the track. We're also looking to make couple of safe spaces in the LGA, so namely our nursery where we grow and develop our colonies. We hope to have that as a safe space where we can avoid there being any baiting there, and that can be a little sort of I suppose little haven where we can keep, at least if the worst came to the worst, we will be able to keep a significant number of colonies there, so that the program would keep going, even if we did have to deal with a red zone. Sorry, Peter says, please speak more slowly when you read questions. I apologise. I'm a fast speaker. I will make a conscious effort to speak more slowly. Right. So Helena asks, why do we still move bees to pollinate farms? It seems like a bad idea. I suppose it's easy to say in retrospect that blue bandits do buzz pollination. Again, easy to say. No, Helena, you're right. I mean, I think that often the problem is that it boils down to economics, and we've kind of really focused on the economics and not really focused on the environmental and knock-on effects of these sort of behaviours. And you're right. I mean, you know, but also on the flip side, because of the way we farm and because of the types of things that we farm, our current practices do rely on the honeybee to pollinate them. If we were to remove all the honeybees, we simply wouldn't be able to replace them adequately. Right? In time, with the regeneration of habitat and a respect and an understanding for the value of our native species, you know, our nearly 2,000 native species, right? We would be able to augment, if not fully replace, the honeybee, but it's just simply not something we can do right now. It's a mistake that we've made and we have to live with, and it's actually quite a difficult thing to fix. So unfortunately, so it would be lovely to be like, right, let's just not move any. We simply can't do it at this stage. Jacqueline Williams says, I've seen honeybees being sold on Facebook Marketplace. Most of the time the location isn't shown, but a number provided. Should they come with a DPI certificate before being purchased? So if they're within New South Wales, then yes, they don't necessarily have to provide you with it, but you should certainly you want to know where they're coming from, because currently we've got very restricted movements on honeybees. You certainly shouldn't be bringing them in from interstate. That is not allowed either. So if they are for sale and they're within the blue zone that we discussed earlier, then you can still move colonies around, but you have to have a DPI permit to allow you to do it. All right, so if that's something that you're thinking about doing, you could. If you happen to be in Ku-ring-gai in the purple zone, then you can't anyway, because you're not allowed to bring hives into that purple zone. Peter says, how far south can you move the hives? Is it too cold down there? That's a good question. So you can't really go any further south than Nowra. I do know of a few populations down in Eurobodalla, but again, that's not that far from Nowra, and it thins. So if you think about Sydney, right, it's the bulk of Sydney, as we move south, their range gets closer and closer and closer to the coast because it stays that bit warmer there during the winter. So it's going to narrow. So you could easily go down to Wollongong, for example, if there was something down there. You probably wouldn't want to go, I would say somewhere like Berry is probably about as inland as you want to go, because any further than that, it's going to get a bit cold. And if you are taking it down south, make sure you've got them at the lower altitudes, because you don't want to have those cold winter temperatures. Yeah, so Helena asks again, basically, why do we still use commercial hives transported long distances when, one, it seems a bit of a biosecurity risk, and two, our native bees are good at pollinating in ways Apis mellifera are not. You're right, and again, I think my previous answer to the reason why I move hives is relevant here. So yes, if we had thought a bit more about it, then that's where we would be, because we would have established and looked after our native populations of bees, and we could certainly, have some local movement of honeybee hives from local beekeepers to do some heavy pollinating in certain industries that perhaps need them a bit more. We wouldn't have to ship them all over the place. The problem is we haven't done that. And you can't, unlike the honeybee that you keep in this little box, most of our native bees, they require native habitat and native plant life to survive. And if you're in an area where you've wiped all that out, it takes years, decades, longer sometimes to re-establish these places. And I don't mean to imply that there's no hope there, because there is. And we are becoming, as a nation, far more aware of the need to rely on our indigenous animals and plants. In fact, there's, like you mentioned, Helena, there's so many of them that are so much better, right? But it's taken us a very, very long time to move away from, I suppose, and we still very much have a Western mindset, but this idea that, perhaps, we should stop trying to be quite so, I don't know, European or North American, and try to be a bit more Australian and do what we can with what we have natively here, because actually it can do just a really good job. So yeah, look, I really hope it's something we go to in the future, and I think we can if we have the right mindset. But currently we're not quite there yet. Right, well, that's all of the questions. So thank you, because there was lots and lots of questions, which is great, because I like to, it means I know you're engaging, because I can't see you all. Look, if anyone has any more worries, then look, please feel free to email me or give me a phone call. Please be assured that I'm all over it. I'm very, very, obviously, especially with the native stingless bees in the program, they are, well, I'm very connected to them. I spend a lot of time with them, and they're very important to me. So I want to make sure that they're safe, and I'll do everything I can to make sure they are. And look, this is very much a community effort. If we all pitch together, we can make a difference on this, even if it's only in our local area. But because we have local bees, in our stingless bees, then that's actually very, very important. So if there's no more questions from anybody, I'm going to wrap it up there. But thank you so much for attending. There'll be links available to this so that you can watch it again if you need to. There's the phone number if you need to call the DPI. I mean, look, to be perfectly honest, pardon me, a quick Google of Varroa DPI will find you the web page you need. It will be the one right at the top. So it's not very hard to find. And yes, if you are worried about anything that's to do with the native bees, especially in relation to this, please get in touch, and if I can help, I will. So have a good evening, everybody. Look, it's been a bit of a pessimistic topic. I get it, right? But look, there is hope, and it's not here yet, and it may not come. So let's be prepared for the worst, but hope for the best. Have a good evening, and I'll see you again whenever I do my next talk. All right. Thank you very much.